Abscission is the shedding of various parts of an organism, such as the plant dropping a leaf, fruit, flower, or seed. In zoology, abscission is the intentional shedding of a body part, such as the shedding of a claw, husk, or the autotomy of a tail to evade a predator. In mycology, it is the liberation of a fungal spore. In cell biology, abscission refers to the separation of two daughter cells at the completion of cytokinesis. In plants function, a plant will abscisse a part either to discard a member that is no longer necessary, such as a leaf during autumn, or a flower following fertilization, or for the purposes of reproduction. Most deciduous plants drop the leaves by abscission before winter, whereas evergreen plants continuously abscisse the leaves. Another form of abscission is fruit drop, when a plant abscisses fruit while still immature, in order to conserve resources needed to bring the remaining fruit to maturity. If a leaf is damaged, a plant may also abscisse it to conserve water photosynthetic efficiency, depending on the costs to the plant as a whole. The abscission layer is a greenish-grayish color. Abscission can also occur in premature leaves as a means of plant defense. Premature leaf abscission has been shown to occur in response to infestation by gall aphids. By abscising leaves that have been made host to aphid galls, plants have been shown to massively diminish the pest population, as 98% of aphids in abscised galls died. The abscission is selective, and the chance of dropping leaves increases as the number of galls increase. A leaf with 3 or more galls was 4 times more likely to abscisse than a leaf with 1, and 20 times as likely to be dropped as a leaf without any galls. Mechanisms. Structural in deciduous trees. An abscission zone, also called the separation zone, is formed at the base of the petiole. It is composed of a top layer that has cells with weak walls, and a bottom layer that expands in the autumn, breaking the weak walls of the cells in the top layer. This allows the leaf to be shed. Lack of chlorophyll as a trigger. The reduction of chlorophyll production in leaves due to decreased sunlight in the autumn explains why some leaves turn yellow. However, the yellow color can attract aphids, so some trees turn the leaves red instead by injecting a bright pigment. The loss of chlorophyll may also contribute to the abscission process. Chemical. A variety of reactive oxygen species are generated by plants during times of stress, including UV light, cool temperatures, excessive light, pathogens, parasites, and high salinity. The presence and continuous production of these ROS causes disruption in the homeostasis of the cellular components, leading to metabolic dysfunction and expression of cell wall degrading enzymes. Hormonal. While researchers originally believed abscisic acid to be the hormone that stimulates abscission, it was later proven that it does not play a primary role. In fact, auxin, a plant hormone, and ethylene have been implicated as prominent regulators of abscission signaling. The two compounds work in a synergistic fashion. As the auxin levels decrease, the flux of auxin to the abscission zone is reduced. Exhaustion of auxin makes the abscission zone sensitive to ethylene. When the plant is then exposed to ethylene, gene expression of cell wall degrading enzymes such as cellulase and polygylacturonase are activated. However, this is not to say that ethylene directly activates WDE gene expression, because the elements responsible for detecting ethylene have not been found in the gene's promoter region. Dwindling auxin levels have also been implicated in autumn leaf color change. In animals,